Thanks, Susanna, and thanks everyone for inviting me to come here and tell you about our work in the Great Lakes. So I realize that this is a quite international audience, and so I figured I'd go to Wikipedia and help you all if you don't know what the Great Lakes are. And they've made this helpful page that clarifies that the Great Lakes are a collection of large lakes in eastern North America, and those are the ones I'm going to talk about today. I'm not going to talk about the African Great Lakes, the Perth wetlands, or the Iowa Great Lakes. I don't know what those are, but they don't hold nearly as much water, I'm sure, as the Laurentian Great Lakes. And these are called the Laurentian Great Lakes because they were formed by the retreat of the Laurentian ice sheet, which carved out these massive basins that then filled in with water about 10,000 years ago that we now know as the Great Lakes. So lakes have been studied for a really long time, and we know a whole lot about lakes that are about this size. This is, in fact, some really well-studied lakes in northern Wisconsin. But the lakes that I'm going to talk about are quite different in a lot of respects, and I would argue that what we know from these lakes doesn't necessarily translate to these lakes. And just for scale here, you can see that you have to zoom out quite a bit before you can capture this entire ecosystem. So what's so great about these lakes? The Great Lakes hold 20% of Earth's surface fresh water. Wrap your mind around that. That's a whole lot of the liquid water that's not salty on our planet. They provide drinking water for over 40 million people, and at the deepest point in Lake Superior, it's more than 400 meters deep. And that means that there's a vast expanse of really cold, dark, fresh water, and we don't know hardly anything about what happens specifically with the microbes in this deep, cold, dark water. These lakes are also really interesting compared to other lakes because they have a relatively small drainage basin. And we think of lakes as being hotspots for carbon cycling, and that's because they funnel a whole lot of terrestrial carbon and organic nutrients into this reservoir where a lot of carbon processing happens. But the Great Lakes actually have a relatively small basin, and what basin they do have is made up of glacial till and sandy soils that's not particularly organic rich or plant rich. And as a result, they don't get quite as much input of terrestrial nutrients as a lot of our typical lakes that we think of. And so they tend to be extremely oligotrophic. In particular, Lake Superior has productivity that rivals the Sargasso Sea. For you oceanographers, you'll appreciate that this is really, really low productivity, and Lake Superior falls in that realm. And so in some ways, these are lakes. In some ways, they resemble the oceans by their size and by their nutrient properties. And so we think this is a really interesting system, and obviously one that's important from a social and economic perspective. We think the Great Lakes are also a really powerful system for studying microbial ecology. And that's because these lakes are all connected hydrologically. A drop of water that falls in Lake Superior, in principle, can flow all the way downstream through Huron and Michigan. It sort of sloshes back and forth between them here into Lake Erie, and then over this big drop we know as Niagara Falls. Fortunately, in the boat, we go through the Welland Canal instead, into Lake Ontario, and then out the St. Lawrence into the oceans. And so microbes could, in principle, disperse from the upper lakes all the way through the lower lakes. But they're a really interesting system because they're geochemically distinct as well. And so over here, we're plotting a few geochemical and physical parameters of these lakes. So temperature varies dramatically from really cold Lake Superior to really warm bathtub Lake Erie. Productivity is distinctly higher in the lower lakes, Lake Erie and Lake Ontario. Phosphorus is really limiting, especially in the upper lakes, Lake Superior. And nitrogen is really interesting, and I won't have time to talk much about it today, but nitrate is actually higher in the upper lakes than in the lower lakes. And so together, this whole fingerprint, I think, makes it a really unique and powerful system that we're just starting to delve into. Okay, and the Great Lakes are also experiencing really rapid environmental change. And I think the time is now for us to really develop time series to watch the progression of these changes and be able to predict what's coming next. So the first change I'm going to talk about, because it affects microbes perhaps, involves nutrients. And starting in about the mid-1970s, phosphorus loading actually started declining in the Great Lakes and many other lakes in the world, thanks to regulations stemming from the Clean Water Act and the Great Lakes Water Quality Agreement in 1978. And so as a result, we don't have phosphates in our detergents anymore. And phosphorus loading has declined steadily since the 70s. But we did not regulate nitrogen inputs at the same time. And as a result, the N to P ratio has basically been steadily increasing in much of the Great Lakes since actually before the 1970s, earlier in the 20th century. And it's not just a result of this loading. There's probably indirect effects that we don't really understand yet that have to do with changes in denitrification and nitrification. And we have no idea what this really means for the microbial communities. This dotted line down here for reference is the canonical Redfield ratio. And so points above the Redfield ratio, we infer that the communities are probably P limited. And if you look at today out here, we're way, way above that line. Here in Lake Superior and Lake Ontario, for example, indicating there's really severe phosphorus limitation in these Great Lakes. But I want to contrast that with Lake Erie, where there's been a somewhat different trajectory. So phosphorus loading declined in Erie for a while, but there's evidence that in recent years, this only goes out to 2010, but if it were to go a little bit longer, there's been increases in phosphorus loading in recent years in Lake Erie, and this is one of the really dramatic consequences here. You may remember in 2014, there was the Toledo water crisis, and the drinking water looked like, well, the drinking water, fortunately, didn't look like pea soup, but Lake Erie water looked like pea soup, and you couldn't drink it because the toxin microcystin was making it into drinking water. And so what we've got here is a situation where the upper lakes and Ontario, to some extent, are actually becoming more oligotrophic, while Erie is becoming more eutrophic. And so again, we've got some really nice contrasts in this system, even though all of this water is connected. Another change that's really, really important has to do with invasive species. And in particular, it's this guy, the Quagga mussel, that's the culprit. And this mussel is only about the size of your thumbnail. And it arrived in the Great Lakes from Eastern Europe in the 1990s in ballast water in ships. There's a lot of shipping that goes through the Great Lakes. And a lot of invasive species around the world are transported by ballast water. And these maps show mussel biomass starting in the mid-90s through 2010. And over a period of just 10 years, these mussels went from barely detectable to blanketing the bottom of Lake Michigan. Today in Lake Michigan, there are often over 10,000 of these mussels in just a square meter of the benthic seafloor or lake floor. And these mussels are really voracious filter feeders. These mussels can filter the entire volume of Lake Michigan in about 5 days. And as a consequence, below this is plotted chlorophyll in the spring. So this is April over that same time period. And what used to be a really nice spring bloom of diatoms and other phytoplankton has essentially disappeared in Lake Michigan thanks to these mussels. And this is quantified here in both chlorophyll and primary production, and those have declined by over 80% since the 1980s, thanks to the mussels and in part to the phosphorus loading declines that we just talked about. But it hasn't affected all taxa in the same way. And as I mentioned, the spring bloom has really been affected. So spring has dropped a lot, while summer productivity has not so much. And the diatoms have been removed, but the cyanobacteria have not been removed. And as a result, the cyanobacteria are an increasingly important part of the community. What used to be a diverse community of eukaryotic phytoplankton is now really dominated by cyanobacteria, including Synechococcus. And I'm going to talk more about this in a few minutes. So you might think that increasing water clarity here would be a good thing. But if you're a fisherman, this is obviously a bad thing. And it also is a bad thing because greater light penetration to the bottom allows benthic algae to proliferate. And so there's some noxious species, including this one called Cladophora, that have taken off in Lake Michigan, in part because of this increased water transparency due to these mussels. And finally, one more change that we should definitely highlight here is that lakes around the world are generally warming faster than the air. This paper a couple years ago highlighted that, and Lake Superior stands out as one that's warming especially rapidly. This map here shows the ice cover duration trend over the past 40 years. And we are losing several weeks worth of ice cover in Lake Superior. This is days per year that have been lost, and this is on the order of 30 or 40 in Lake Superior on average. And as a consequence, summer surface water temperatures have gone up. And so we're getting much warmer summer surface temperatures. Lake Michigan this year was about 8 degrees above normal on the surface. Great for swimming, but consequences for the rest of the ecosystem, of course, to be determined. So given these changes, we think that it's really important that we start to develop a time series in this system, and we've started to do just that. I was surprised when I started this work 6 years ago that there was virtually no data on microbes in the Great Lakes. To me, they're really familiar. I live in Chicago, and so I look at Lake Michigan almost every day. And I think a lot of us take them for granted. They're pretty familiar. But from a microbial perspective, we know so little about them. So starting in 2012, I was lucky enough to team up with the US EPA's Chicago office, and they've been running water quality surveys in the Great Lakes for over 30 years. But they never ever looked for bacteria, archaea, viruses. They didn't look at anything smaller than phytoplankton. And so we were lucky enough to get on this cruise on the Lake Guardian. And I should mention here that this infrastructure obviously is much different than what's been used for small lakes in the past. And it's telling that Great Lakes studies at NSF fall under the purview of oceanography, because this is the kind of ship that you need to do this work. And so we go out with them in April and in August, and the entire cruise takes almost a month. We start in Milwaukee, and we do all of these stations here shown on the map. So we do Michigan and Huron first. We switch off in Detroit, and then we do Erie and Ontario through the Welland Canal. And then we come back and do Superior last, which gives us a few more weeks for the ice to disappear in April. And our team has been sampling 4 depths in particular that you'll see in my coming slides. Surface up here in the summer. If you're not accustomed to thinking about water column profiles, we get thermal stratification where we get a thin layer of warm water sitting on top of a thermocline, and then cold water, essentially 4 degrees all the way down to the bottom. So we sample this surface mixed layer, this deep chlorophyll maximum that often occurs just below the thermocline, the mid-hypolimnion, and then 10 meters off the bottom, B10. And so we had to address some really basic questions to start because there was so little data. We wanted to know how unique the Great Lakes were compared to other freshwater and potentially even marine systems, how the Great Lakes differed from each other, what made them really unique, lake by lake. And I'm going to focus today on a couple key taxa, the deep water ones that really distinguish the Great Lakes, and these cyanobacteria that are becoming increasingly important. So I'm not going to spend too much time on this, but my postdoc, Sarah Paver, has really spearheaded a lot of our Great Lakes work, and she led this meta-analysis of about 2016 studies to ask where do the Great Lakes fall on the continuum of freshwater and marine systems. And it turns out that her results uphold this very classic salty versus fresh divide. We've known for a long time that salinity is one of the major drivers that distinguishes microbial communities, and we see that here, though perhaps the Great Lakes are a bit distinct from other freshwater systems. If you're used to thinking about freshwater, a lot of the most abundant taxa will seem very familiar to you. The most abundant things are actinobacteria, beta proteobacteria, and this SAR11-like alpha proteobacterium. And these bugs are cosmopolitan. They are found in all of our lakes, and they're really abundant everywhere they're found. And so these are really the dominant ubiquitous guys. But then we see a number of interesting taxa like this one. The fourth most abundant taxon in our entire data set is this chloroflexi organism that's poorly studied and only shows up in the deep water. And so we were really interested in these deep water taxa because these shallow lakes that have been really well studied for a long time don't encompass this habitat. They don't have this really deep, cold, oligotrophic water. And so we actually found a whole number of taxa shown here that are enriched in this bottom water relative to the surface. And many of these are really poorly characterized. So the chloroflexi really stand out as the most abundant, notably not in Erie. But we also find, for instance, marine group 1 archaea, so ammonia oxidizing archaea, a lot of nitrifiers, nitrospira, nitrosomonads, and a number of really poorly characterized groups down here. So the metabolism of these organisms is not very well understood, and I'm happy to say that we are now pursuing this in collaboration with JGI through a CSP that we were just awarded, and we're hoping to sequence some SAGs from some of these deep water guys and try to reconstruct their metabolism. So when we start to look across our entire data set across the Great Lakes and ask, what patterns do we see that, perhaps not surprisingly, depth is the strongest driver of community similarity. And this very much echoes what we see in the oceans. So in the summertime, that's all these circles. These are the surface communities. We see the DCL communities sort of in the middle, and the hypolimnian communities off to the right here. And this axis is collapsed down here into bars, so you can see the overlap. And what's kind of cool is that we see that spring samples, as these crosses here, essentially look like the bottom water during the summer. And that's actually not surprising, because in the spring, this stratification disappears. We get isothermal mixing all the way through the water column. And since the bulk of this water is this deep water, this is the signal that we see even in the surface in the spring when the water column is mixed. But when you zoom into just the surface water, you start to see that each lake has its own unique fingerprint. And so this PCOA here shows a very clear distinction between the upper lakes, Superior, Michigan, and Huron, and the lower lakes, Erie and Ontario. And that nicely reflects the differences we know from the environment. The temperature tends to be warmer in Erie and Ontario, and they're far more productive in terms of chlorophyll. And we think temperature in particular is really important in driving this pattern. You can see, particularly in Erie and Ontario, the crosses here from 2014 and 2015 are distinct from these triangles. And that separates warm years and cold years. 2012 and 2013 were warm years. 2014 and 15 were much colder years, polar vortex years if you lived in the upper Midwest. And so we think temperature is really a key factor. And in the circles here, we actually see that a colder year in Lake Erie and Ontario looks more like a warmer year in a cold lake. And so again, we think that temperature is going to be really key in understanding these patterns. So that was all 16S that I showed you. But we've now sequenced about 24 metagenomes, and we see similar patterns unfold when we look at short read similarity via K-mer similarity here in a heat map. And so this is from about 20 metagenomes, and you see this big group here is the deep water in the deeper lakes. So Michigan, Huron, Superior, and even Ontario when it's stratified. And they share a lot of sequence similarity. This group here is the summer samples in these upper lakes. And the summer surface samples are really distinct because that's where all the phototrophy is happening, and it's a lot warmer. So that makes sense to us. Erie and Ontario have distinct surface communities and then distinct deep communities as well. And so when we sat back and looked at these results, it makes a lot of sense because when you're out on the boat, this is what the water looks like in Erie, and this is what it looks like in Superior. And so from this, we can conclude that there's pretty strong evidence that depth and that lake are really important drivers of microbial communities across this system. So now I want to move on to a genome-centric approach. And can we actually reconstruct genomes in this environment and start to look at individual taxa and at metabolic pathways? And this work I'm going to show is the work of Justin Podowski, who is a graduate student who's over there somewhere. So you should talk to him if you have some more questions. And what Justin did is assemble 21 metagenomes individually, and that'll be important in a minute, and bin the contigs by composition and coverage, and then map all of our short reads back to those mags to look at distributions. And this is what he found. We've essentially created a genome atlas of the Great Lakes now that encompasses 570 genome bins, greater than 70% complete. And importantly, these genomes recruit back about 50% of the short reads. And so we think we're capturing quite a substantial fraction of the dominant microbes in this community. And so, of course, this tree has a lot of information, and you could spend all day just clicking through it and finding interesting things. But just to highlight, we've assembled some ammonia-oxidizing archaea, cyanobacteria, and chloroflexi, the usual players in freshwater, actinobacteria, beta proteobacteria, and alpha proteobacteria. But there's a whole lot of patterns in here that we're just beginning to mine. So I want to zoom in on just a couple examples. And this one I sort of randomly chose, the gamma proteobacteria. And I should mention, these bars show read recruitment colored by lake. And so of all the reads that recruited to this particular genome, all of them came from Lake Erie. But there's a cluster here. And what's cool is that we recovered essentially the same exact mag from 3 different samples. And it is cosmopolitan. It recruits reads from all 5 of the Great Lakes. But then sister to that, we see this clade that appears to be specific to Erie or Erie in Ontario. And this group down here is interesting because we find it abundant in all of the lakes except Lake Erie. And so this read mapping is really powerful for showing us patterns of ecological variation in these genomes. So the chloroflexi I mentioned are really important in the deep water. And we recovered about a dozen genomes that, again, have over 99% average nucleotide identity, really similar to each other. But we recovered them independently from about 12 different samples. And the read mapping is very consistent across them. Again, tells us they're almost identical genomes. And we find these chloroflexi in every lake except Erie, and not at all in the surface. These 2 depths here, the blue and the green, represent the hypolimnion depths. So these chloroflexi are really preferring deep water and deep lakes. In our final example, the LD12 group, and this is the freshwater sister to the SAR11 group that you may have heard about. Again, Justin recovered about 20 genomes of these LD12s, and they're really, really closely related. Almost no branch length here, and over 99% average nucleotide identity. But you can see a little bit of potential clustering, different populations, as inferred from variation in read mapping. This first group, for instance, recruited a little bit more from Superior, and in fact, those mags were actually assembled from Superior samples. Okay, so I want to switch gears and talk about cyanobacteria because, as I mentioned, they are becoming increasingly important. And as Susanna mentioned, my PhD was in Penny Chisholm's lab, so picocyanobacteria are near and dear to my heart. And so, of course, I'm really excited that there are lots of Senecacoccus in the Great Lakes. And so this is just a oligotype heat map for 16S. And so this showed us that there are a whole diversity of oligotypes of cyanobacteria in the Senecacoccus family, some of which are cosmopolitan across all of our samples. Some seem to be really specialized for the upper lakes, and some seem to be specialized for the lower lakes. And so, again, we're seeing some early indications of ecological diversity. And over our time series, we can see interesting dynamics for some of those oligotypes as well. Here we see this distinction between the warm years and the cold years, where one of these oligotypes swapped abundance or dominance. And the great thing about cyanobacteria is that they're really easy to separate phenotypically using flow cytometry. They have their own tags in the form of pigments. And so by flow cytometry, we can see that there are distinct populations in all of our samples. This superior sample, for instance, has a population that lacks phycoerythrin, as well as 2 similar populations that have abundant phycoerythrin in addition to chlorophyll A. And so in collaboration with Rex and Danielle, we've sorted and sequenced individual cells from populations like this and sequenced whole genomes to try to look at genome diversity in these groups. And in addition to those SAGs, we've also reconstructed about a dozen genomes from metagenomes, assembled those. And so together here, I'm showing our current picture of the cyanobacterial genome diversity across the Great Lakes. And so if we start with this heat map here, the sorted cells from Rex fall into groups 1 and 2 here, these big groups. And then we've got some MAGs that add a little bit more diversity here and recruit abundant reads as well. And so we've now named 5 groups, group GL1 through GL5, shown here in this heat map. And they're shown here in this tree for context with marine and freshwater cyanobacteria. And what's kind of cool is that the Great Lakes cyanobacteria and freshwater cyanobacteria in general don't form a distinct clade that's separate from marine strains. So marine and freshwater strains are sort of mixed together in this group down here. Here we have the really abundant Prochlorococcus and marine Synechococcus, but some of these are marine strains as well, 5701, RCC307, and some Chesapeake Bay strains. And so our groups fall across this tree. So GL1 through 5 are all across this tree. And what's cool if you've looked at cyanobacteria is they're really, really pretty. So here's some colors of marine cyanobacterial strains. And we've found from our genome predictions that these Great Lakes cyanobacteria encompass all of these pigment types, and so we expect them to be all these different colors, and that their distribution will map on to where that light harvesting is optimal. And so we can map distributions through read mapping like this, and we can start to assign some ecological characteristics to these 5 groups. And so we find that some groups are specific for the upper lakes and some to the lower lakes. And we're trying to understand now what the genomic basis of that differentiation is. OK, so my final bit I would like to end with is moving beyond sequencing to look at activity of these populations. And to do that, we're using quantitative proteomics. And this is a method called DIDO-IPTL that was developed by my close collaborator, Jake Waldbauer, at the University of Chicago. And the gist of this method is that you use isotope labeling to label the peptides in two samples, and then you run them through your mass spectrometer, and you essentially quantify the relative abundance of isotope 1 and isotope 2 to get an indication of peptide abundance. And so this is really inexpensive, but more importantly, it's really sensitive and precise, not only for culture work, but also for metaproteomics. And so we applied this to what would normally be a pretty boring set of samples in the spring when it's completely mixed from the surface to the bottom. And by DNA, the communities are not very different between the surface and the bottom. But we were curious whether we could see differential activity at the protein level, even though the composition of the community was pretty similar. And in fact, we could. And so this is a heat map of protein abundance in the surface versus the bottom, and you see these clear, distinct groups. And the outlier here is a surface sample from the summer, which we expect to be quite different. And what you can do is look at the relative activity of each taxonomic group using this protein expression metric. And so to the right here, we have fold change increasing at the surface, and to the left, fold change increasing in the deep sample. And we find, perhaps not surprisingly, that the cyanobacteria were much more well represented in the proteomes in the surface, suggesting higher activity, while these nitrifying groups and the chloroflexi were much more active, as inferred from proteomics, in the deep samples. And so we think this is a really powerful approach to look at activity, not only of taxa, but of pathways across our ecosystem. So just to conclude, hopefully I've convinced you that these lakes are indeed great, that they harbor novel phylogenetic and genomic diversity, that ecological distributions point to there being clear distinctions between the deep lakes and the shallow lakes, and Erie is pretty much an outlier by all metrics. Justin's recovered over 500 genome bins that reveal surprisingly low diversity in the LD12s and the chloroflexi. The cyanos are becoming increasingly important, and those, by contrast, are really diverse, not only genomically, but phenotypically, and we're following up with cultured isolates now in the lab. And we think that activity can be precisely quantified by proteomics, and we're excited about doing some more of that. So I'd like to thank the captain and crew of the Lake Guardian and the EPA who've allowed this sampling to happen. From my lab, it would not be possible without my postdoc, Sarah Paver, and my graduate student, Justin Podowski, who's here. The proteomics was all done with Jake Waldbauer and Lichun Zhang, my collaborator, Ryan Newton, Rex, and Danielle at JGI, and thank you for your attention.